Now comes the fun part. The next thing I want to do is take a piece of paper here, and I'm going to actually make a picture using all the parts of the flower. So I'm going to take the pistil right here, or most of the pistil. I've got the stigma there, and right at the top, and I've got the style, and I'm going to take a piece of tape, and I'm going to tape that down right in the middle of the page so it doesn't move. All right? Then I'm going to take my better forceps here, and I'm going to take the part of the ovary that's somewhat still intact. That's that little piece there, and I'm going to tape that down as well. Okay, next, let's position the stamens. So I've got a stamen over here. Let's just lay them out and see how it would look. I'm going to put the stamens sort of like this. Alright, so I got 2. Let's put another one there. That's 3, 4, 5, whoops, 5, and 6. How does that look? Oh, let's try to get them right in the middle there. Okay, that's kind of cool. Move that one a bit. Okay, I'm going to take one piece of tape right across all of them at their base. There we go. I think I'll put one more just to really hold them down. Now we're actually going to take a little piece of this in a little while. That's not a big enough piece. Let's go for another one. Right across the whole thing. As I said, we're going to take one of those stamens. We're going to knock off some of the pollen, and I'm going to use that microscope and show you it up close. It looks really cool. Alright, so we're getting there. We've got our central female part, that we've got the pistil with the stigma, the style, the ovary. We've got the male part, which is called the stamen. This is the anther, this is the filament, and of course, we need to add some petals. And the petals, the function of the petals are, well, to protect these are the sex organs, right? So to protect the sex organs, which are essentially open to the environment, as opposed to the cone, which I've got over here. Alright, so might just put a couple of those petals there. Take a piece of tape, let's move that one out of the way. Tape this one down. Looks alright. Take this one. Oh yeah, and the other thing I wanted to say about the petals, of course, aside from protection, these things have nice colors, right? We've got some nice yellow leaves here to attract pollinators, the insects and the animals that come along and feed on the plant and pick up the pollen and then go to another flower and fertilize it by depositing the pollen. So I'm just going to tape that one down as well. I think I'm just going to stick with the 2 petals so that it doesn't get too cluttered. I can come back here, and really, this is the base of the ovary as well. I mean, I've got a little bit of the ovary here, a little bit of the ovary there, so I might take this and stick that on there, which has got part of the stem, so that we understand that's part of the flower as well. Oh, and I just ran out of tape, so let's take that piece and keep that one for later. Tape that guy down there. There you go, that looks pretty nice. Now if you wanted to, you could actually label this thing. You could take your pencil, put a line out here, you could label that, you could label this part here, grab that part, label that, label the filament, right? You could come over here and label a petal, you could come over here and label the stem, that kind of thing. So make a really nice little picture. Here's another flower that I've got as well. It's another lily. Have a look at this one. Completely different color, but we've got all of the same sex organs. Right? We've got a slightly different shaped stigma here on the top, but this is the female part, and these are the anthers. So we're going to have a look at some pollen now under the microscope. So let's do that. I've taken one of the anthers off of that red plant, red flower, and I'm now trying to knock off some of the pollen into this little plastic dish. So I'm going to have just pollen in this one. All right, and there's a lot of it. I don't know how well it'll show up, but we're going to hope for that. All right, put that down there. So I've got pollen in this one, and I've got actually a full anther in that one. So we're going to have a look at those under the microscope. So I've taken some pictures with the microscope, and some of them turned out better than others. Here we can see the anther, which is the male reproductive part, and it's under the 10 times objective here, and it's just this rod-shaped part right here, and all of this brownish, orange stuff is the pollen. So let's isolate the pollen. What I did is I tapped the anther. You can see a few pollen grains here. So let's see another image. So this is again on the lowest power, and you can see that's the pollen in a little dish. Let's zoom in a bit, and now you can see this is the anther again with the pollen still attached. As we've zoomed in, we're under the 60 times objective lens, and all these bean-shaped structures are the pollen. So that is what you would see. If you were to look really, really close at the anther of a flower, you would see this just packed with pollen, and this again is the male sex cell or gamete. This is the stuff that flies through the air or gets attached to insects, which then travel from flower to flower, and it gets deposited onto this structure. This is the stigma under the low power objective, and you can see there is a little bit of pollen already on it. If we were to zoom in, which I'll do in a moment, the pollen sticks onto here and creates a tube, travels down this part. This part here is the style. This is the stigma. This is the style, and the pollen would travel down all the way down to the ovary where the eggs would be. Now we've really zoomed in 60 times onto the stigma, and you can see that it's got all these projections here, and these are sort of like think of it like Velcro that helps the pollen stick to it. It's a really sticky surface that helps the pollen adhere. Now we're looking at a leaf under 60 times, and you're starting to be able to see these areas, these little pockets. These are the actual leaf cells, and the light is shining up through the leaf in this case. This was the yellow lily leaf that I had before, and all these things are the pollen grains. And finally, if we zoom into 200 times, we can see these are leaf cells. They look very sort of liquidy, translucent because they are still alive. And these are pollen grains which have stuck to the leaf. Alright, so I hope you enjoyed this little video. Hope to see you in the next one.